Have you ever thought that is it possible that there would be a human that would be made artificially once? And yes, that is what I'm talking about in this complete scenario or in this complete session. That is the life that had been assembled inside the world's first cell that had been built from the scratch. And ultimately, this is the artificial life that we are talking about. Yes, you have heard this correctly. We will be talking about this in this session along with me. My name is Bhavar Singh Churuhi. I'm ex-NDA 115 course and qualified civil services in 2012. And I welcome you on the platform. Now over here, when it comes to the complete news, there is the cell that had been formed with no living ancestor and it just grew despite or copied with the copied its DNA and then it divided as well. Now, this complete scenario, it happened at the University of Minnesota, and this research was led by synthetic biologist Kate Adamala. She announced the SpudCell, that is a cell-like system built piece by piece from the non-living chemical components with no natural cell that had been used anywhere in its construction. Now, this cell actually, it fed, it grew and replicated its own DNA and divided, completing a complete basic life cycle or cell cycle for the first time right from the scratch. Now, this preprint had been released through the Biotech, and again, this is a very important fiat that we are talking about. Now, also over here, it is very important that this cell has just about 90 kilo base pairs that are there. Now, when it comes to the base pairs are basically the DNA material that is there, which is required for sustaining the complete cell. And previously, it was thought that the minimum criteria for a cell to sustain itself is 113 kbp or 113 kilo base pairs that are required. So over here, this is very important that this is the smallest functional synthetic genome ever assembled, built from just the chemicals and not copied from the nature as well. So over here, when it comes to the scientist, she had doubted herself after the result. And what happened was that Kate Adamala, who is the synthetic biologist at the University of Minnesota, she had spent years trying to build a cell just without borrowing any of the substance from the living one. Now, when her teams finally divided under the microscope, her first reaction was disbelief. Obviously, that would be the reaction if the same had been done by you as well. And she double and triple checked the data before going public. Now over here, when it comes to the lab, there is a small academic team that is working with Biotech. Now Biotech is basically the non-profit Adamala had co-founded, and they chose to release the result as an open preprint rather than wait for the peer review cycle as well. Now, the method that they have used is not genetic modification of any bacterium. There is no genetic material that has been taken from any bacterium, but it is a liposome, that is a lab-made fat bubble, which had been assembled around the synthetic DNA and enzymes with nothing borrowed from the existing organisms. And also, there is the outside voice as well. We have the origin of life scientist Jack Szostak, and he is involved in the complete work. He has called this as a rare instance of an artificial cell effort progressing this far, a signal that the field takes very seriously. So this is the thing that we'll be talking about. We'll be going in and we'll be doing a complete deep dive in of the topic. So be with me. But before moving ahead, let me tell you that we have the GS Foundation batches on the platform of StudyIQ. Now you can enroll in these batches because they are based upon the SMART model, and we are starting off with a new batch. We have already started. The admissions have already started. The first class will be live from the 7th of July. So you can enroll into the batch by using the code BRSLive. That will fetch you the maximum discount. Now, this is the complete timeline. Now, when it comes to the first, it was in 1953 where the Miller-Urey experiment was conducted. Now, they had simulated the early, earth chemistry to show that the simple organic molecules which are there, that are the life's law ingredients, they could form spontaneously. Now, this was in 1953 in the Miller-Urey experiment that was conducted. Now, in 2000, we had the first synthetic viral genome where the scientists, they chemically synthesized the nearly about 9,600 base genome of the hepatitis C, which was synthetic genetics that moves from the theory to the bench. Now, later on, it was J. Craig Venter in 2010, which had synthesized the genome as well. And ultimately, this was a bacterial genome that we are talking about and transplanted it into the existing emptied bacterial cell. And this was called SYN 1.0. Then we had in 2016 where we had the SYN 3.0. That is the minimal genome, where there was a step down 531,000 base pair and 473 gene genome, the smallest genome known to support the autonomous replication that was done in 2016. But over here, it is very important that this was basically top to bottom approach. Top to bottom approach where the materials, they were synthesized from the living bacterium and then they were transplanted. So this was basically top to down approach, but this is where we are talking about bottom to up. That is, we have only the chemical constituents which had been used for the process of completely synthesizing a cell from the chemical constituents. Now, from 2023-24, there was a global coordination also that began, and we had the Asian Synthetic Cell Initiative, or SynCell Asia, launched. And over here, this was joined by the existing European Synthetic Cell Initiative and the US-based Build a Cell Network as well. And this is what we are talking about, the SpudCell, which had been built from the scratch, and it is the first time that we have the cell-like system assembled from the non-living parts alone. So this is very important to note over here that this is actually not a new organism. This is just a new construction method that we are talking about, where the earlier synthetic cells, Syn 1.0 or Syn 3.0, they were basically impressed. They'd be impressed by writing the DNA on a computer and inserting it into the existing bacterium's body. Over here, the SpudCell break that lineage. That is, for the first time, we are seeing that the container, the genome, and the protein making system, they were all assembled from the chemical parts with zero contribution from the pre-existing living cell. Now, over here, it is very important that there is no natural donor cell that was used at any stage of the construction. The genome capacity record had been broken, where it is at the 90 kilobase pairs, which beats the prior assumed 113 kilobase pair of the floor. And there is completed a full basic cell cycle where it had fed, then it has grown. There is the DNA replication that was done, and then there was the division as well. And this had been released as an open preprint before the formal peer review. That is a governance and verification question in itself as well. Now, over here, it is very important that this has not been classified as a living organism by any of the definition or any scientific definition, because actually it cannot survive itself. All right, this is very important to note over here that this cell, it cannot survive on itself, and that is why this is not a living cell that we are talking about, because it requires constant external delivery of the ribosomes and the nutrients as well. And that is why this is not a living cell right now. But over here, this is about the essential paradox. Over here, in the words of Dr. Kate Adamala, she had said, we have to replicate it. We have replicated in chemistry what only used to be possible in biology. It proves that the most fundamental functions of the life do not need a mysterious magical spark. But over here, this is about a tension between the mechanism as compared to vitalism. Because ultimately, if life can be written as a software code, that is DNA, and then it can be booted up inside and assembled chemical chases, so is life is merely an advanced biological operating system? That is the question that it raises. So over here, it is very important that this is just beyond the obvious political, economical, social grid that we are talking about. This is about the origin of life science itself, where it is a working bottom-up cell. All right, this is bottom-up cell where the synthesis has been done by the chemical constituent itself. So this gives the origin of RIBE researchers a lab bench so that they can test how actually the simple chemistry could have crossed into biology billions of years ago. Because we know that the complex organisms, they developed from the chemicals itself. Then also this is about the definitional crisis, where the SpudCell is deliberately built to be understandable component by component. But at the same time, it still resists a clean yes or no answer to whether it is alive, exposing how thin our working definition of life actually is. All right. And then again, it is about the biomanufacturing frontier as well, where we have now a fully synthetic chassis that could someday perform the chemistry that natural cells cannot. And ultimately, there will be the new routes to the medicine, materials, and carbon capture without farming a living organism at all. And this also deals with the containment paradox, because ultimately when it comes to such kind of living organisms, they poses a threat as well. But when it comes to its dependence, this is on the lab supplied ribosome, and it is the biggest safety feature when it comes to this cell, because it cannot survive outside the strict lab condition. And that built-in fragility that this cell has right now, it is not an external rule, but it is what currently prevents its misuse as well. All right, so this is very important because when it comes to such kind of scenarios or the genetic modification, so we have the different governance matrix for the biosynthesis in India as well. We have GEAC in India, that is Genetic Engineering Appraisal Committee under the Ministry of Environment, Forest and Climate Change, which is apex regulatory authority evaluating the environmental releases of any synthetic or the engineered organisms. We also have the Department of Biotechnology, which is under Ministry of Science and Technology, which actually formulates the biosafety guidelines and manages the review process through the Review Committee on Genetic Manipulation. And then we have the global accords as well. We have Nagoya and Cartagena Protocol that are under the CBD, which governs the fair biological resource sharing and cross-border safety of living modified organisms as well. So these are the governance metrics when it comes to the different species that could be created or they could be genetically modified. Now, this is very important, but ultimately this shows us that what the wonders of science they can do. And ultimately, this synthesis of the cell from just the chemical components, this actually shows, or it is very pertinent because ultimately this leads us to how the life that began on the surface of the earth. We have the various foundation batches which are there on the platform of StudyIQ. For English, we have the Resolution P2I batch, and these GS foundation batches, they are modeled upon the SMART model of framework, where you get the structured classes, you get mentorship, assessment, revision, and test. Now, over here, it is very important that these batches are important for those who are planning to appear for 2026 or 28 examination, or even if you are planning to appear in 2029. So you can build your base and ultimately you can have the guidance through the best of the teachers which are available for the civil services. Now, while enrolling into the batch, let me tell you that you can fetch the maximum discount. And for that discount, you have to use the code BRSLive. That is BRSLive. That is there. That will give you the maximum discount. So that would be all in this session. But do not forget to like the session, to share this session with your friends, and also to subscribe so that you do not miss any of the updates in the future. Thank you. Bye-bye. Take care and Jai Hind.